Number 5, epigenetics and cancer treatment. For decades, scientists and oncologists assumed that cancer was caused only by damage to the DNA sequence of critical genes within cancer cells. But recently, a more complex picture has emerged, and it is one that shows that some cancers are caused by epigenetic changes, which are alterations in the environment around the DNA that impact DNA packaging and gene expression. Unlike genetic damage to DNA, epigenetic changes can be reversed with treatments and can be less toxic than conventional chemotherapy. For example, in brain malignancies such as glioblastomas, scientists are now studying samples from patients' tumors to understand how normal epigenetic states are disrupted in the tumors and during the treatment process. Emerging research suggests that protein affecting epigenetic states are critical biomarkers and drug targets. Such knowledge of wiring differences in the packaging of DNA within cancer cells could help doctors one day prescribe more potent and precise drugs that hone in on specific epigenetic targets within tumors.
And our expert is Dr. Johnathan Whetstine, Associate Professor at the Mass General Cancer Center and a leading expert on trying to drug the epigenome. And the question for you, Johnathan, is how do you get at specificity given how global these epigenetic mechanisms are? Do you need to combine drugs, and do you have to target individual genes? What does the future look like for specificity?
Thanks, Daniel. It's a really important thing to realize that there's a complex interplay that happens within the organization of your DNA. So when we talk epigenetics, I think it's really important to realize that this is how your DNA is packaged and organized. And what the cell has inside there is a repertoire of components, whether it be proteins or RNAs that were alluded to yesterday, that are very important in making sure that organization is in place. As we begin to understand this more and more carefully, we realize that each individual component plays a key role in different aspects, whether it be regulating genes involved in oncology or immunology, or whether it's the organization and how the genome is stabilized or destabilized. And these things are very specialized to make sure this massive process takes place. And for that reason, the beauty of the epigenome, or regulating or harnessing the control of the DNA packaging, is that you can impact certain processes, whether it be oncogenic genes or it be the amplification of drug-resistant targets. So what you can do is you would combine that potentially with therapies so that you can accentuate those attributes, or better said, if you know that mutations are occurring in factors affecting your genetic stability, you could then alter your therapy and not give something that would be less effective. And so what I would say about the epigenome is, as we begin to target it, yes, it's not just global. We've begun to realize that there are very specific attributes, and that really the power that's going to be emerging over the future is understanding the proper combinations. Where can you use it in a very deliberative way to strike the Achilles heel in a cancer cell, yet sustain a happy individual? And so I think that kind of combination and realization that there's various components controlling the package, the expression, the stability, the fates, we can control and target those in a very unique way. So it is a bit more specific than I think we realized because of the skill sets of the proteins or the RNAs involved. And if we target them and biomark them, we can begin to become more and more specific and combine it in the right place.